Welcome back, everybody. So we're into the last couple parts of our module on nucleic acid structure, DNA replication, and chromosome structure. So what we're going to look at today is the first part of nucleic acid structure. We're going to go pretty quickly, so let's get started. When we think about DNA, we characteristically think about the double helix, which is kind of synonymous with the structure of DNA for most people. But we're going to have to go a little bit deeper into the structure of the molecule so that we can fully understand its functions. Let's start off with a broad overview. When we think about the structure of DNA, we think about it having multiple levels, and then some of those levels extend up to the level of chromosomes, which we'll look at a little bit later. So the most basic level of structure we can think of is the individual nucleotide. So up here we've got some individual nucleotides, and those are polymerized together using condensation chemistry. We talked about that before. We basically link two monomers here, nucleotides together, using a reaction which releases a water molecule. And that gives us single strands of DNA. So here's a single strand of DNA, and then two of those strands wind together to form a double helix. And we'll talk about how you get that structure when we talk about DNA replication. And then the next level of structure we can talk about is taking the double helix and looping it around some proteins. And this gives us a structure called chromatin. And these proteins here are called histones. So the double helix is wound around these proteins called histones. And so DNA isn't just naked. This is a naked DNA helix. And then in cells, we see the DNA usually wrapped around proteins to form structures called nucleosomes. And those proteins are specifically called histones. And then there's some additional levels of DNA structure, which we're going to skip over here and come back to later. And ultimately, we wrap all of this DNA and these histones up into structures we call chromosomes. And that's the structure we see in the nucleus of cells. There's slight variations if we look at what goes on in prokaryotes, and we're not going to get into that. We're going to focus on eukaryotic DNA structure in terms of chromosomes in a little bit. So let's start off with the basics of nucleotide structure. So this is a DNA nucleotide, and I know that because the base has been labeled as deoxyribose. So we'll come back to that in a moment, but let's just start with look at the overall structure. There are three parts to any nucleotide. So let me get my pen just to mark this up really quick. The three parts are the DNA base, sometimes called the nitrogenous base. We'll come back to that in a moment. And then there is a sugar molecule, and here that sugar molecule is deoxyribose, and we'll compare that to the structure of the sugar in an RNA nucleotide in a moment. And then lastly, there is one or more phosphate groups, and here we're just seeing one. It could be 1, 2, or 3. In the actual DNA molecule or an RNA molecule, we'll only ever see one phosphate molecule. So let's come back down to the deoxyribose here. DNA is called DNA because it has deoxyribose in it. It is deoxyribose nucleic acid. And so the deoxy part of this name comes from this carbon here, which is labeled the 2' carbon. So let's first address why these carbons are numbered 1', 2', 3', 4', and 5'. That is because chemists have numbered the nitrogens and the carbons in the base 1, 2, 3, 4, 5, and so on. And so if we want to talk about the carbons in the sugar in the nucleotide, we need some way of discriminating between the carbons here and the carbons in the base. And so they are given this label of 1' or 2' or 3'. The prime just lets us discriminate between, it's just a dash, it lets us discriminate between the carbons in the sugar and the carbons and nitrogens we find in the base. So the important part about the deoxyribose nucleotide is that the 2' carbon only has this hydrogen here. So this hydrogen down here identifies this sugar as deoxyribose. And we'll compare that to what we see in an RNA nucleotide in a moment. So if we look at the 4 different types of DNA nucleotide, the 4 different types of DNA nucleotide vary based on the base. And so you're familiar with these. We have adenine, which base pairs with thymine according to the rules, and guanine base pairs with cytosine. And what you see is that the double ring structures, 2 rings, there's 1 ring here, and then this is a ring also. And so these double ring structures are called the purines, adenine and guanine, and the pyrimidines have these single ring structures, thymine and cytosine. Now, the important thing is that adenine and thymine are going to form base pairs, and they'll form 2 hydrogen bonds between them, and cytosine and guanine, when they base pair in the DNA, they're going to form 3 hydrogen bonds. So we always have a double ring structure base pairing with a single ring structure. What that does is it keeps the 2 strands of DNA parallel. If we put the 2 double rings together and the 2 single rings together, the 2 double rings would give you a helix of this width, and then the single rings would be narrower, and so the double helix would be doing this, but the 2 strands run parallel because we actually have a double ring structure like adenine base pairing with a single ring structure like thymine. So those are the 4 DNA nucleotides: deoxyribose containing adenine, thymine, guanine, or cytosine. Okay, so let's take a look at the RNA nucleotides, and let's just review the structure of the nucleotide again. We're always going to have this base, and the base is going to be attached to the 1' carbon of the sugar. Here is our sugar, and then here is our phosphate group. So it is important for you to be able to describe how this is assembled, and ideally how to draw this. So you should be able to tell me the base is connected to the 1' carbon, and then the phosphate is attached to the 5' carbon over here on the left as it's drawn. And remember, hey Hetty, how you doing? Remember, these things are drawn typically like this, but you may be asked to recognize these structures if they are drawn upside down, reflected, that kind of thing. So make sure you're familiar with the chemical numbering. That means you don't have to worry about memorizing lots of different orientations of the nucleotide. Okay, let's take a look at the 2' carbon in ribose. So the RNA nucleotides contain ribose, and what you see here is that the 2' carbon has a hydroxyl instead of just a hydrogen. So deoxyribose doesn't have this oxygen here, and so it's called deoxyribose because it lacks that oxygen. Whereas in an RNA nucleotide, you see this 2' hydroxyl. The only other thing I'm going to mention here and introduce about the structure is that the 3' carbon is going to become very, very important when we talk about DNA replication. So I've talked about the 2' carbon here and how you can use that to discriminate between ribose and deoxyribose. The 3' carbon with this hydroxyl group is going to become very important when we talk about joining nucleotides together. So let's look at the 4 bases that we see in RNA nucleotides. RNA, like DNA, has adenine, it has guanine, and it has cytosine. But what you see is that the thymine that you see in DNA is replaced with uracil. So if you see a chain of nucleotides, if you see a nucleic acid that contains uracil, you know that it's RNA. If the chain of nucleotides has this 2' hydroxyl, you know it's RNA. So make sure you can discriminate between the 2 types of nucleic acids. Sorry, let me back up really quickly. Based on the different types of bases you see, and on this 2' carbon structure in ribose versus deoxyribose. Okay, so let's move on to talk really quickly about the polymerization of the nucleotides and how that leads to some directionality. And so I'm going to use a word I've used before, and that's polarity, but I'm using it in a different sense. When we talked about polarity before, we were talking about electrical charge differences that gives us things like polarity in water. So when I'm talking about polarity here in this molecule, rather than talking about charge differences, I'm just using polarity to describe the fact there are 2 chemically distinct ends, much like the planet is polar in that it's got 2 discrete poles. Here we've got 2 discrete ends. So let me grab my pen real quick. Okay, so when we link these nucleotides together, we can see here we've linked together a thymine-containing nucleotide, one with adenine, cytosine, and guanine. So these 4 nucleotides have been joined together. That's what DNA replication will do, that we'll look at in a little bit. So what we're going to do to link these together is we're going to take the hydroxyl that was on the 3' carbon here, and we're going to join it to the phosphate group. And so we do a condensation reaction between those 2 groups, and we get this bond which involves this phosphate here and that becomes known as a phosphodiester linkage. And so that's the special name given to the covalent bond that forms involving phosphate between two nucleotides. So when you link these nucleotides together, they always get linked together, moving in a 5' to 3' direction. So what does that mean? Remember, the carbons are numbered, and so there is the 5' carbon. So if we were linking nucleotides together, we would add the next one, and it would be adenine, and then cytosine and guanine. So if I want to add another nucleotide, I have to add it here to this 3' hydroxyl. And so the next nucleotide would be joined in at this end down here, what we see as the bottom. And so the linkages are made 5' to 3' as we join the nucleotides together. Is what this leaves us with is a phosphate attached to the 5' carbon at this end of the molecule, and so that's called the 5' phosphate, or just the 5' end. And then the last nucleotide has the 3' hydroxyl down here, and so that's sometimes called the 3' hydroxyl end, or just the 3' end. So that is the chemical polarity that we see in DNA. A strand of DNA is always said to have polarity, and it runs from 5' to 3', and that usually refers to the chemical nature of each of the ends. Now we'll come back to that when we look at DNA replication, and it becomes very, very important in terms of thinking about DNA replication. Okay, so if we take two strands of DNA and we wrap them around one another, what happens is we get the double helix, the characteristic double helix. And so the bases here always point in. This was the seminal work of Crick and Watson. So here are the bases pointing to them inside of the helix. And we've got these hydrogen bonds which link the two strands of DNA together. Remember, hydrogen bonds are weak electrostatic interactions. They're individually weak, but when you've got thousands, hundreds of thousands of these in a DNA molecule, they hold the two strands together. So if we want to break these bonds and pull the two strands apart, we've got to put some energy in, we've got to do some work. And so the act of pulling two strands of DNA apart to initiate DNA replication is going to involve some work, and so that's going to be a challenge we have to come across a little bit later. The distance between the nucleotides, the width of a nucleotide is about 0.34 nanometers, not too important right now. There are 10 nucleotides per turn, so a turn of the double helix is from the top of this strand here, if we trace it all the way around to when we see the top of the strand again. So that's one turn. There are 10 nucleotides, 0.34 nanometers for each nucleotide. A complete turn of the double helix is 3.4 nanometers. So if you knew how many bases there were, or how many nucleotides there were in a piece of DNA, you could work out how many turns. If you knew how many turns there were, you could work out how many bases and nucleotides. If you knew how long a piece of DNA is, you could work out, therefore, the number of turns, and therefore the number of nucleotides in a piece of DNA. So those are some interconversions that it's useful for you to be able to do. The bases point inwards, like I said, and then these red strands, a little hard for me to trace because of my red pen, these red strands are the sugar-phosphate backbone, and they wrap around one another, forming the characteristic double helix. And so they are this part of the molecule here, and so the sugar-phosphate backbones are called the sugar-phosphate backbones because they alternate phosphate linked to sugar, linked to phosphate, linked to sugar, linked to phosphate. And so when we join these nucleotides together, we get this repeating structure of phosphate, sugar, and that gives us a structure called the sugar-phosphate backbone. And like we just described, one strand will have a 5' end, and the other strand will have a 3' end. Let me back up real quick. Is what you just saw, and the reason I did that is so that I cleared my notes. Okay, so we see that this strand on the left has a 5' end, and this strand on the right has a 3' end, and so that would be the directionality of 5' to 3' running top to bottom as we're looking at the diagram. In the double helix, the other strand, so the other strand is over here on the left, the other strand always runs in the opposite direction. So the 5' end is at the bottom as we're looking at it, and the 3' end is up here at the top. And so that direction of that strand is running from bottom to top as we look at it. And so this is one of the important characteristics of DNA. the two strands are kept equal distances apart because we pair a purine with a pyrimidine, and the two strands of DNA run in opposite directions. So this gives the double helix its anti-parallel nature. So the parallel nature means the strands are kept the same distance apart, like railway tracks are kept the same distance apart. The anti-parallel part comes from this chemical polarity that one strand runs 5' to 3', and the other strand runs in the opposite direction. Now this can become really important and somewhat problematic when we look at the mechanics of DNA replication. So I will wrap up there, and when we come back next time, we'll start looking at the mechanics of DNA replication. So I will see you very shortly.